might have seen the announcement. The topic is on harnessing high-performance computing across STEM disciplines. So I will request our panelists to present what HPC, what use of HPC in their respective fields are. So I will be sharing their slides that are with me, and we can go from there. Let me share my screen. Can you see the screen? Okay. So let me see. The first one is Alec.
Yep, so I'll get started. So yeah, so I'm going to just talk about ways in which we use computation for machine learning fluid dynamics. So the motivation for this work is that large amounts of energy are lost due to turbulent drag. 5% of all man-made CO2 is from turbulent drag. And so it's become common to use machine learning with high-resolution fluid simulations in order to try and mitigate drag, among many other sorts of tasks that we're interested in. And so the three sorts of types of computation that I'm going to discuss are how we actually run high-resolution simulations to get data for fluid dynamics. Here in the bottom left, I'm just showing an image of a plane wing where you can see there's these very small scale vertical structures. And then I'm going to talk about how we can take these simulations and generate low dimensional models by training neural networks. And then the last thing I'm going to discuss is how you can incorporate these various things for tasks like reinforcement learning to do things like minimize drag. So can you go to the next slide? Sorry, can you go to the next slide? Has it updated? Yep, there we go. Okay, sorry, yeah, one back. I think my computer is just moving a little slow. Okay, so first, let me just talk about the direct numerical simulations. Sorry, slide 4. Okay, so these simulations can take on the orders of days to weeks to months. So here on the left, I'm just showing you one of the simplest fluid simulations that exhibits turbulence. This is called Couette flow. In this simulation, the top wall is moving to the right, the bottom wall is moving to the left, and the colors that you're seeing here are just the velocity moving in this X direction, left to right. And even for this simulation, which is basically the smallest simulation where you can see turbulence, you can see there's these chaotic dynamics. You have this low speed fluid at the wall highlighted in blue that breaks up. And running this simulation still took 100,000 grid points. I ran it on 8 processors, and it took about 2 days. If you try and run a larger simulation, like I'm showing you here on the right, which is a 3-dimensional airfoil, what you're seeing on the right here are just isosurfaces of vortices. A simulation like this can require 25 million grid points. This one took 220 processors over 2 weeks in order to get a sufficient amount of data. And so these simulations are very high dimensional and take a long time to run. And usually we use low-level languages like C++, Fortran in order to run these sorts of things. Can you go to the next slide, slide 5? And one way that we can use the data from these high-resolution simulations is in machine learning tasks. And so the first task that I'm going to mention is you can use machine learning for low-dimensional modeling. And so a typical approach to generate low-dimensional models of fluid flows is to do something like I've shown here on the left, where you can take U here would be our high-resolution image. You can encode it using an autoencoder to a lower dimensional state representation. And then you can evolve it forward in time with a neural network I've written as a neural ODE here And then you can decode it, expand the dimension back out into the high resolution simulation dimension. And so, whereas in the case of running the high resolution simulations, we're doing it, we're solving the Navier-Stokes equations. Here, the computational task is to train a set of neural networks with all of this data that we generated from the high resolution simulations. And just to illustrate how much dimension reduction you can get, here on the top right, I'm showing the high resolution simulation, and below it, I'm showing a model that we were able to train with only 15 degrees of freedom. And you can see qualitatively, they look quite similar. And if you compute the statistics, they're in excellent agreement. And so, so we can build these very low dimensional models also. And then can you go to the next slide? So then the last thing I want to talk about is sort of the problems that we face in reinforcement learning. So finally, I want to talk about reinforcement learning. In reinforcement learning, the goal is to find a set of actions on our fluid simulations to reduce drag. So typically in reinforcement learning, you feed an agent information about your state, the simulation at some time. It'll give you an action. That action then goes into your environment, which often is a high resolution simulation, which gives you a new state and a reward. This new state then goes back into your agent, gives you a new action, and this process happens iteratively. And in this iterative process, you train a neural network, which is the agent, to learn the best set of actions to maximize your reward. And so I've been mentioning there's this high computational cost for these high resolution simulations. Well, what we found in our work is that if you generate models, like I presented on the previous slide, these low dimensional models, you can mitigate much of this high computational cost by just doing it on these low dimensional models. And here at the right, I'm just showing you an example of control we learned on a low dimensional model applied to a high resolution simulation. The control you're seeing are just some synthetic jet actuators that shoot up and down with the red and blue at the bottom. And this curve that you're seeing is just the streamwise velocity. So when it flattens, that means it's become laminar. So this is one way we can speed up reinforcement learning. And then can you go to the next slide? The final way that we can speed up reinforcement learning that I want to discuss is we can also just train many of these environments in parallel. And this allows us to basically have like embarrassing parallelization where we just train many, many agents together on many different processors. And here on the bottom right, I'm just showing you an example of where we did this to flow around a cylinder where the cylinder rotates and with the goal of reducing drag. And you can see that the wake behind the cylinder starts out short and eventually elongates as it's reduced drag. And so those are the three things I wanted to talk about, just the expense of high resolution simulations, how we can build low dimensional models, and then ways that we can apply reinforcement learning. And with that, I'll pass it on to the next person. Thanks.
Okay. So I'll just continue. So I'm going to talk about HP's high performance computing in biological and biomedical sciences, and like iterating like why we need like computing resource for biology area. I'll go from more about genomics that we the most consuming data we are looking for, we are facing right now. Can we go next? So it's just a basic a concept about the reference genome. So you may know DNA, like for every organism, we have a set of DNA short for deoxyribonucleic acid. So a reference genome is the sequence database assembled in a like a representative for a specific species. For example, like this year, like 2023 is the 20th anniversary for the first, since the first human genome was assembled and released. And as of today, human genome has evolved into a more wide versions, like the Pangenome project is completed this year as well. So so you can see here based on the, so like DNA is basically composed by like 4 letters like A, C, G, T. And some part of the genome, they are repetitive and we cannot know specifically what exact sequence they are. So there will be like ambiguous bases in every assembled genomes. So for like human genome size is about 3 gigs or so. So that is like for 1 copy of the genome. And every data we get, we can like reference for this reference genome to look into if there's a mutation, there's a variations that's important for biological processes, for example, disease, for cancers, for any things that might happen. So next. So in addition to the one we faced very often is the human one. It's like 3 gigs. You would know from every other species, especially in a plant, they can go to 17 gigs and even more with the wide variety of species. Like for wheat, it's like really big reference genome. And I know for some trees, people studying forests, they have troubles like how to assemble such big genomes for their reference species. Okay, next. So so I think like since 2000 years or so, there's a techniques called high throughput sequencing that developed, and until today, you can see on the left side. So it's an example from Illumina. I think it's next 6,000. So right now they can output like 6,000 gigs base per run. So this run will finish in like about 1 to 2 days for just 1 machine in 1 lane. So you will know like how many data people are generating now. So once we got this short reads, so which are randomly short, like 100 to 200 base pairs, then we can map or align these reads to the reference genome we just talked about. And then we call this mapping or alignment. And we can, once we pile up the reads, we can know which specific locations we got variant, like we show in the plot on the right-hand side. And so that's why like we apply HPC in genomic data analysis. Okay, next slide. So currently I just try to summarize like most common types of omics data we're getting now. So the example I just showed is the DNA sequence. So it's basically we can refer to like genomics. So there's also different variations for genomic sequencing, like we got whole genome sequencing. We got whole exome sequencing. We just sequence the genomic variations in exons. So you can see on the right-hand side, like exon is the coding regions that will actually transcribe into RNA and translate into protein and function in a biological, in a creatures or in organisms. And also there's variations like ChIP6, short for chromatin immunopresentations. They are trying to target specifically for the protein and DNA binding loci. Also on the right-hand side, you will see a term called transcription factors. So they are proteins that will bind to DNA. The regions we call promoters, and they can directly lead to the downstream gene expression. So this is the genomic sequence we're getting now. And we also get transcriptomics that's specifically sequence for RNA transcript. And currently, I think is really outburst data is the single cell RNA-seq. So we can get like 10,000 cells, their transcriptome at just 1 run. So that's for transcriptomics. So can we go next? Another type is the epigenomics that is also very important. So you can see ChIP-seq for histone modification is one type of the proteins that will be entangled with DNA. So basically the concept is the same as ChIP-seq, but they are looking for epigenomic features. So they won't change the DNA sequence, but rather modify the genomic or architecture for the DNA. Another type, for example, is for bisulfite sequencing. So we're looking for one epigenetic marks called DNA methylations. So you can see on the right-hand side, so we got ACGT in our DNA sequence. Methylation means we add one more methyl group to the cytosine on the fifth carbon of the cytosine. So we use chemicals, sodium bisulfite, to treat this, treat the DNAs. So there will be, you will see like the methylated cytosines won't change to any features, but if there's not protected by methylation, that will directly change into thymine. So by these comparisons, we can know which specific loci is methylation modified. And epigenomics is also really important because they won't change the DNA sequence, but they will directly lead to the gene expression differences. Okay, so next, please. So we just go through like the main genomic data as we might got. So I just want to go through like the basic computational approaches we are doing like every day now. We are starting from the pre-processing, like from the sequencers we just showed from Illumina. They will produce short reads and we do alignment to the reference genome and we do matrix assembly. So for the downstream analysis. And then next we'll do focus analysis. We call differential expressions. We call genomic variation. We might find biomarker so based on GWAS studies, and we can do mathematical modelings to predict outcome for this individuals, like disease predictions. And the final steps, so we would do data visualizations. You will see the top one is like we call sequence logos. They're basically using probability models to show the ATCG occurrence for specific positions. We might create heat maps to show differential express, differential methylation. We might also see feature plot, like for specific cells, for specific genes, their expression levels. And we might be able to trace the cell development with techniques like RNA velocity or the pseudo time analysis. So it is basically the genomics approach we have right now. And also all these procedures, they will need software or algorithm development. So that's the bioinformaticians doing, they do software development for all of the process we need for the genomic data. Okay, so I'll go let it to the next person.
The next one is Robert.
Hello, everybody. So I think we're a little bit running behind on time, so I'm going to try to give a really big overview of my topic. I am going to give another talk in the upcoming year that's going to dive a bit deeper into this and give a lot more specific ways that you can use, like specific coding that you can actually implement to analyze geoscience and climate data. You can go ahead and go to the first slide. So geoscience is increasingly taking a systems approach. And what do I mean by that? Well, historically speaking, and even to some extent now, people were either like an atmospheric scientist or an oceanographer, or maybe you're a geologist or you studied ice. However, to solve things such as climate change, more and more, you really got to be a jack of all trades. So you really have to understand human interactions with the Earth system, as well as all of these different processes. And as Alec alluded, with some of the fluid dynamics that occurs, especially geofluid dynamics, that can be very computationally heavy. Go ahead and go to the next slide. over the last few decades, the global observation system has become increasingly advanced. So historically speaking, we would have more in situ or like on the ground type of measurements. You would either have things such as weather balloons, or you would have a stationary place point gathering data. But more increasingly, we have so many different types of satellites that are consistently measuring the planet and circumvent the planet every day, taking the same measurement over and over and over again. There's also ship data, so people go out in ships and collect data, and as well as having ocean buoys and just aircraft that fly around collecting data. Next slide, please. So, for example, reanalysis data, which is from the European Center for Medium Range Weather Forecast, takes all of this data and they fill in the gaps that the data may have through climate models. And one such of the variables or outputs that the ERA5 puts is temperature, precipitation, wind speed, and direction. And they do this at hourly intervals for every 10 by 10 kilometers around the globe. So you can imagine just the size of the data being collected every single hour being outputted. It's thousands of petabytes of data, in fact. Next slide, please. You can skip this one. I thought I deleted it. And so really this data though interact with each other. So it's not even just one layer of data. So you have your longitudinal and your latitudinal data, but then you also have vertical exchanges. So the different layers of the atmosphere going as high into as like the boundary layer between the troposphere and the stratosphere. You also have ocean data that goes pretty far deep into the ocean. And the other dimension that's not shown here is time. So for example, a current project that I'm working on right now takes daily data from 1979 to 2020 and has all these other spatial components to it. So the data sizes are massive. And you can go to the next slide. So Earth system models then take all of these data and combine with all the different Earth processes. And these are obviously simplified Earth processes because no computer in the world is powerful enough to model every single process accurately. So we take reasonable assumptions about the geophysics that occur, and we simplify everything. And the models basically take all this information and make projections. And that's kind of what you see on the left. And what you're seeing on the right are basically different outputs of these data. And every single one of these outputs, so one line of data, depending on the temporal and spatial resolution, can be a few terabytes in size. And you can go to the next slide. Now, luckily, there is amazing software out there. For example, the primary tools that I use to analyze the data are MATLAB and Python. And I started off in MATLAB, and I really like the syntax of MATLAB a lot better. But MATLAB kind of requires you to load the data into memory. Versus Python, you can do what's called lazy loading, particularly through X-ray. And this is fantastic because even if you have terabytes of data, for example, when I was a researcher at the Jet Propulsion Laboratory, we had about 40 petabytes of data. And obviously, even with my brand new top of the line Apple computer right now, I only have 64 gigabytes of RAM on this. So my computer wouldn't be able to load all that data. So you're able to analyze the data and build out your code through lazy loading. You're able to kind of see some of the outputs. But in order for you to actually run all of that data and visualize the data, so plot the data, you're going to have to send it to an HPC because once you actually hit the compute button or try to plot it, everything gets loaded into memory. And so you need to really compartmentalize the data and send it out to a high performance computer. And more, due to the different sizes of the data, geosciences have been moving to cloud computing. So because the data sizes can be in the terabytes or petabytes even. Um, Places like Amazon Web Services is a great place to host the data. And then you can use TangioForge to connect the website and either download the data if it's small enough onto your computer, or more importantly, you can kind of tinker with the data and do calculations on it until it's a reasonable size where you can actually bring that down into memory. Next slide. So just a very quick example of how this would happen is, so for example, the Lawrence Livermore National Lab, as well as the Department of Energy, host some of these climate models. And so we can access these models, and we'll, if you go into the website, you'll see these different parameters that have like the frequency, your resolution that you want it in, as well as the variable that you're interested in. Next slide. And you can kind of just build out some Python code. And it's not important that you understand this code, but basically you kind of gather what you need from the website, connect to the website virtually, and then you can either download, like these ones are very small bits of data, so I was able to download these onto my machine. But if you go to the next slide, you can also just connect to this data virtually. So this is one project where I needed to collect to the data virtually, and I needed to request 100 cores. And this is using this was before I was a postdoc at UCLA, so this is actually using Rutgers University's HPCs. And here I requested 100 cores, and each core was about 15 gigabytes, and this took a few days to run to actually output this. So you're basically able to like access a huge amount of data, do some calculations on it, and then something that's more in the gigabyte size. Once you get it down through all the calculations and you get your output that's about a gigabyte or whatever size that you actually think that is reasonable and have the stuff that you're most interested in, you can then download that into your computer and have that handy to like put in your paper, or just have like in memory for future use. So it's a pretty powerful tool because you don't actually need the data or computational resources locally, and can just do all this virtually. But as I said before, I will be taking a deeper dive into how to utilize this and giving more concrete examples of like certain Python packages that allows all this to happen in a later presentation. Thank you.
This is Namjoon.
Oh, I prepared a separate slide. Can I share it with the audiences?
Oh, okay. So let me.
So so in this talk, I'd like to briefly discuss about an idea on how parallel computing can be applied in diffusion model. So this talk will be very brief. So I'll be mainly focusing on explaining overall ideas of diffusion model very briefly, and we'll also explain in which step it takes so much computing resources, and how the parallel computing can be used to overcome that computational burden. So diffusion model is now replacing generative adversarial network framework in generative modeling, in modern machine learning, for many data modalities, including image, audio, or even text. So this model is specifically well known for its super good sample qualities for the newly generated data, as you can see in this slide. This is the overall framework of the diffusion model. So it is basically a latent variable model comprised of two stages. So first, the first step is about the noising step, and like, let's say that we have a data of the dog, and you perturb the picture of the dog by adding some very small Gaussian noise for like capital T times until the original noise turns into a pure noise. And the second step is about like denoising step, tracing back the data perturbation processes. So usually you start from the pure Gaussian noise, and by tracing back the perturbation processes, you generate a new sample out of it at the end of the iteration. So nice observation of theory of diffusion model is if you perturb the data using very specific form, say the stochastic differential equation, then the reversing step can be derived with a score function of the marginal probability density, like denoted as a PTX at noising level T. And we estimate the main goal of the diffusion model is to estimate this score function. And we usually do that through some very sophisticatedly designed neural networks, such as a UNEC. Then once we estimate this score function through neural network, we can, using this reverse stochastic differential equation, we can generate a new sample, with a estimated score function. So that was like overall idea, very brief idea of the diffusion model. And so very simple method for sampling is to discretize this reverse stochastic differential equation. Usually you update the sample X, which is supposed to be a pure Gaussian noise at its initial stage, capital at time capital T, and you get a new sample out of at the end of the iterations of this recursive update. And another very nice theory of diffusion model is if you have the following reverse SDE, then there always exists a corresponding ODE that has the same solution path with that of the reverse SDE. So if you look at this so this is a solution path, the squeaky line, the solution path of the SDE, and there always exists a corresponding ODE that has the same solution with this stochastic differential equation, and you can derive the form in this way. This is the form of the ODE that we want to solve. So while solving this ODE or SDE, we need to recursively update this discretization of the differential equation capital T times. And in practice, for generating new image, we usually set this capital T as like from 1,000 to 3,000. And this is the step where, so the sampling is the step that takes so long time, and this is where the parallel computing can play some role. Okay. So recently I found this paper trying to solve the computing issues in sampling for diffusion model through parallel computing. So the key idea is to use Pickard iteration, which is an iterative method used to approximate solutions for solving certain types of functional equations. So let's say that we have this ODE with a initial value, initial condition, X naught. Then like the analytical solution of this ODE can be written as follows. We just move this dt onto the right-hand side and take the integral from 0 to t. And we have this form. But the challenging task of solving this solution is to solve this integral form, right? And the idea of Pickard iteration is, let's say the initial condition as the initial guess for the solution, and you put the initial condition onto this XA and solve this integral form and get the first solution of X1. And it's the next guess of the solution, and you put this next gets to another integral, like you put this x1 to this this integral form, and you solve another integral which is usually solvable in general cases, and you do this like capital K times until the solution converges to converges. And although I was not fully able to understand the full the mathematical ideas suggested by this parallel this paper, the key idea is that yeah, we can compute the integral in this. So in order to compute this integral, we first need to discretize this integral into like a summation of t terms, and we can like compute each of the summands like in parallel by using some parallel computing, so that we can like reduce the computational times for the convergence of the solution. So this is a computational graph of the sequential sampling for the naive diffusion sampling sequential sampling in the diffusion model. Yeah, you recursively update these samples. But in here you can create this very specific computational graph with a skip dependencies, and like you only do like the capital K iterations instead of like doing the capital T iteration computations. Yeah. So in practice, this capital K is much smaller than the capital T. So you do this computation by using the parallel computing, and you just only do the whole iteration like capital T capital K times. And this is the table that suggesting that their suggested method gives us like 3 to 4 times speed up while maintaining the qualities of the samples. So that's what I have prepared.
Yeah, great, thank you. The next one is Xiao. You are muted, Xiao. Your mic is okay.
Okay,
Now,
can
yeah,
can you use my screen?
yes, we can see.
Okay. Okay. Here I will talk about how to model interacting dynamic system, and wait. Okay. Okay, and here we can see in an interacting system we have a set of objects. They may interact with each other, and they can show very difficult behavior along the time, and it will generate a dynamic graph. Here we give an example like a molecular dynamic system. We can see they are dynamic, and their interaction is also dynamic. Here, we will talk about how to model this interacting systems. The first tool we use is graph neural network. Here, when the graph structure is given, and our graph neural network can approximate pairwise object interactions, and it can retrieve how the system changed from the time step t to time step t+1. Here, the core of graph neural network is to aggregate information from every node's neighborhood, and to update node representation in a in a recursive manner, and finally we can generate. We can have a very good graph node representation, and finally we can use a decoder to generate to output our state. And the second tool is about neural ODE. I think maybe we have learned something about ODE. Yeah, like it's you can see this formulation. It's a basic ODE. Here the G define the derivative of the node representation, and how to turn an ODE into neural ODE. It's about we can use a neural network to model the derivative of every node representation. So our ODE can be solved by some ODE solver in some black models, in some black box method. And another thing is in dynamic system, we will have some new interactions. We have some new interaction, like some interaction may form and some interaction may drop. They will disappear. And how we can model this complex dynamic system, and our basic framework is to combine our graph neural network with neural ODE, and it can drive the evolution of the dynamic system. First, we will use an encoder. We will use an encoder like MLP. We will infer the initial state of nodes and edges. And the second is we will use some generative ODE. Here we will use graph ODE. We will combine graph and ODE to model the evolution of the dynamic system. And finally, we will use the decoder to reconstruct the observation xi. Like we have some, yeah, we need some type of output, and we will use a decoder to output that. Yeah, it's our basic framework to use graph ODE for modeling dynamic system. Like we have some applications. The very typical application is to model the spread of COVID-19 in the United States, and then here every node is different state, and they will dynamic change. We will have some daily confirmed cases, some days like, and also we can build some edges. It's like the population flow among states, and then we can make some prediction about future state, and we can show our graph ODE framework can model this very smoothly. And also here the graph ODE may have some challenges, because the generated model they may fail to model some complex high order correlation, like their encoder they may miss some high order non-neighborhood semantics embedded in graph spectrum. And the second part is they usually use the first order derivative in the graph ODE formulation, so it cannot capture high order dependencies in latent representation, like some Newton's laws of motion. They can we can say the first order derivatives cannot model this. So here we also give some updated version of the neural ODE. Here we will use the second order ODE solver, and in this way we can model high order dependencies of dynamic system, and we can achieve better performance and efficiency compared with the original one. Yeah, and maybe we can talk about our new frameworks later. Yeah. And it's about this is about some techniques in modeling dynamic system, and also we can see this system can be used in some like physical simulation, like some applications we our fellows talked before. Yeah, so it's very interesting. Yeah. Thank you.
Thank you, everybody. So if people have questions from the audience, please raise your hand and I will let you speak. Okay. One question I got from, oh, Francisca, yes. You need to unmute, Francisca.
Actually, TV, it's me. I'm not sure why I'm listed as Francesca, but Francesca is also here. My question to all of the panelists, are you all using the Hoffman-2 cluster?
Yeah, I've used the Huffman-2 cluster for some pre-processing, and then once the data is more in a reasonable size that I can handle it locally, then I just use my laptop.
And for the others?
I use Hoffman-2 for very large simulations. We have DoD resources also.
Yeah, I will also run my code on some servers, but it's not that large servers. It's just very normal servers.
So for me, I haven't used a Hoffman chip server. I have used like AWS servers from AWS or yeah.
I use Huffman-2 as well. So for preprocessing simulation, if I got a final like matrix or so, I can just use my local computers.
Fair enough. Thank you for sharing that. My final question is, if you could change one thing about the Hoffman-2 cluster, what would it be?
So for me, I like interacting with my data, and so one frustration with Huffman-2 is just the interface where I have to wait for it to run and finish to see whether a certain line of the data is correct. And I could try it on little subsets and do all of that stuff like offline Hoffman-2, but with other HPCs that I've used in the past, you can use like Jupyter Notebook on there directly, so you can kind of go line by line before you execute everything to the HPC.
I wish there was more memory in home and Scratch, and also how frequently Scratch gets replaced. In my old cluster, in my PhD, it was our own, I guess, so we just always had our data on there, and having to move data back and forth is
So
a little bit more of a challenge.
You mean the disk space, not the memory?
I
Yeah.
think
It's not RAM. I mean, yeah, disk
Okay.
space.
I think also the similar thing like you always need to move the data from one server to another server, and yeah, it will cost a lot.
Yeah, I think you can buy memory if I'm correct, but I don't know. It seemed like it wasn't worth it for anyways.
Yeah, you can buy the disk space. That's I think 136 per year per terabyte.
Great. Thank you all for sharing that.
Any other question? So let me ask one question to every one of you. How much, let's say, what's next coming in your particular field? Like what are the future demands in very near future demands of computational power within your field? and type of power too. For example, GPU or any other kind of computation.
If no one else is going to go, I can go. So in my field, more and more things are moving to cloud computing, and there is a lot of data that's already on cloud computing, but it's still compared to the big picture, it's still very limited. So that's like a new area, and also just to speed up how to interact with data that's on the cloud.
There are a lot of people in fluid dynamics. I was just at our big conference in Washington that are very interested in quantum computing. I'm not sure the details on how you incorporate quantum computing into fluid dynamics simulations, but there is a lot of interest in it.
I think now the large language model has been used in like some graph neural network and some dynamic systems. So maybe in the I think in the future I will also try something try something about the large language model, and maybe it will or maybe at that time we will use like a lot of TPUs. I'm not sure. Yeah.
Okay, thank you.
For me, it's like what I present today is basically for the reference-based analysis, genomic analysis. So we got data for those organisms that have references, but now like we are developing alignment-free method to directly go from the data we got and then have models based on the training. Of course, is like a learning-based model to do disease prediction. So that is just not just for computation, but also like we are lack of sources for the biological data as well. But the idea is the alignment-free algorithm we are using to predict disease with epigenetics.
Namjoon.
For me, currently I'm doing research on tabular data generation. So actually, it doesn't require that much computational resources because the tabular data itself has a very low dimensional data structures. But I'm definitely thinking of extending the current methodologies to other data modalities like images or text. So just like Xiao told us, like I'm also thinking about extending. So in the diffusion model, like the transform architecture also can be used, and transformer model takes lots of memories. And but although I'm not working on the transformer right now, in the future I need a lot of computational resources for doing that research. Yeah.
Very good. Any other question from the participants? If not, then it is right time. It's 12:29. So you get back 1 minute of your time. So let's thank you to all the panelists and congratulations to all of them. And we'll see you in next event, which will be sometime in January. So thank you.
Thank you all.
Thanks. Bye.
Thank
Thank
you,
you.
TV.
Thank you.
Bye-bye.
Thank you. Bye.